Let's see this Kreb cycle. What is Kreb cycle? Kreb cycle is also called citric acid cycle because citric acid, this citric acid is also a tricarboxylic acid. That is why this is called tricarboxylic acid cycle. It is an amphibolic pathway. See, this is amphibolic. Why amphibolic pathway? This has function for catabolism and anabolism both. And it is active both in fed condition and fasting condition. So I will first talk to you about what happens in fed condition, how this acetyl coenzyme A converts into CO2. Then I will tell you a little bit about fasting condition also. Okay. So fed condition, that is why this is pathway is called amphibolic pathway. It is associated with catabolic as well as anabolic and active both in fed and fasting condition. So you can remember what is catabolic process related to, associated with, in catabolism is associated with glycolysis. During fed condition, it is associated with glycolysis. During actually, what is the anabolic function? It is associated with synthesis of this glycine. This porphyrin or heme synthesis. For synthesis of fatty acid. During fed condition also, if you take too much of glucose, glucose will convert into fat. So we will talk about fatty acid synthesis. See, everything is related, but you will have to connect many things. I will actually help you, but you will have to memorize the things in the beginning. Now gluconeogenesis, these are the anabolic function of this Kreb cycle. This is catabolic function of Kreb cycle. From glycolysis, production of energy. From catabolism is associated with glycolysis, anabolism is associated with fatty acid synthesis, gluconeogenesis, and porphyrin synthesis. These three pathways associated with catabolism. This is associated with anabolism. Sorry, this is associated with catabolism. These are associated with catabolism. We will talk first about this catabolic processes. Okay, that is associated with glycolysis. So function is actually, first function is to generate NADH from this catabolic process, and then you know this from anabolic process it will help generating fatty acid, glucose, and porphyrin ring for hemoglobin, heme. We actually talk about all this pathway in MD1 only. Now you can see, so this will, in fed condition, it will produce NADH and FADH2 and ATP. So pathway occurs in all pathway occurs in mitochondria, requires oxygen. Without oxygen, it cannot happen. So what will happen? This is the whole reaction. One acetyl coenzyme A will produce 3 NADH and 1 FADH2. Total 4. Reducing equivalent NADH plus FADH, NAD plus FADH2, 4. 3 NADH and 2 FADH. And there will be 1 GTP production, and then these are the energetics. 2 carbon dioxide will be released. So we'll theoretically yield 10 ATP because this is 7.5. This is equal to 1.5. This is 1 ATP. So 7, 8, 9, and this is 10. So 10 ATP. Both carbon of acetyl coenzyme leaves as carbon dioxide. Conversion of isocyt at which places? These places. So we will talk about these places. So here we will end this process. Okay, this we studied conversion of this to pyruvate. As you can see, in glycolysis, glucose converts into pyruvate. Now in this process, the energy is released. Now from we are actually in the process of talking from pyruvate to carbon dioxide. So we have already done how this pyruvate converts into acetyl coenzyme. We have already done how this pyruvate converts into acetyl coenzyme A. The enzyme is called pyruvate dehydrogenase. And we have already done that. So how pyruvate dehydrogenase convert this pyruvate to acetyl coenzyme A through oxidative decarboxylation. Now today we will talk about how this acetyl coenzyme A is converted into this CO2. So you can see, can we compare this metabolic pathway similar to the flow of river?
Yes.
Yes, because you can see in the flow of river, what is happening? This flow of river will rotate the turbine.
It's
Water
like a
wheel.
dam.
Yes?
No, I said it's like a dam, right?
Yes, it's dam. So it will release electrical energy. Okay. Now the same way, can you see here, the energy is flowing from this, and because of this, there is flow of this glucose has high energy, and the water here has high potential energy. The same way carbon dioxide has high kinetic, because carbon dioxide is air. You can see it is mobile, very highly disordered molecule. So you can see high energy, high potential energy to low, high potential to low potential energy means high kinetic energy. So this converts into this because of this flow of this glucose to carbon dioxide. From glucose 6 carbon dioxide is what is produced, NADH and ATP is produced. NADH and ATP is produced. That is, NADH through electron transport chain releases energy. So through this process, what happens is you can compare this whole process is to the flow of river. Conversion of glucose to carbon dioxide to flow of river and release of energy as a result of this. So this has very great implication. We will learn this all metabolic pathway, just consider like a flow of river, especially catabolic pathway. And we will call this catabolic pathway downhill reaction. We will use the terminology downhill reaction. Downhill reaction means the reaction from higher potential energy of molecule to lower potential energy of molecule. And in that process, because of this flow of this higher potential molecule to lower, there is release of energy. That means that so downhill reaction means high energy molecule, high potential energy molecule, as you see water here at high potential is converted into low potential energy, and as a result there will be release of energy. So you have to remember this downhill reaction this way. Downhill means all catabolic reaction. So this, remember this. So this Kreb cycle is not a downhill reaction cycle. So when we talk about cycle, there is a uphill reaction and there is downhill reaction, and that we will talk at last. Okay, first let me talk about this Kreb cycle. At last I will tell you which is uphill reaction and which is downhill reaction. In any cycle there will be uphill and there will be downhill reaction. All cycle is composed of uphill and downhill. We will talk about that uphill and downhill reaction at the last. So let me first talk about Kreb cycle. You know Kreb cycle, after pyruvate enters into this mitochondria, and what happens in mitochondria, pyruvate converts into acetyl coenzyme. You already know this. By which process? Oxidative decarboxylation. Oxidative decarboxylation. How many coenzymes are there? Which all vitamins are present in PDH?
5.
Vitamin B1?
All the way to 5, right? And look.
Yes. 3. And just in place of B4, what you write? Lipoic acid.
Lipoic acid.
And then B5. So what does B5 means?
CoA.
Very good. B1 means TPP.
Thiamine. It's
FAD. B3 means
N.
NAD. Very good. So this is what happens at this process. One carbon dioxide and one NADH. This is a very important reaction. This is called bridge reaction. You call it bridge reaction because glycolysis and Kreb cycle is joined by this reaction. Now acetyl coenzyme A enter this Kreb cycle. Now acetyl coenzyme will enter Kreb cycle, and as a result of Kreb cycle, you will see this acetyl coenzyme has 2 carbon. This 2 carbon will be released as 2 carbon dioxide. So Kreb cycle will release carbon dioxide from this acetyl coenzyme. And in Kreb cycle, there are production of NADH, 3 NADH and 1 FADH2. 3 NADH, 3 NADH and 1 FADH2. Remember this energetics. Now we will talk about the pathway. So rather than talking about pathway, let me talk about enzymes. So we have seen how many NADH is released here? 3 NADH plus 1 FADH. So total how many dehydrogenase steps should be there in the Kreb cycle? 1, 2, 3 and 4. 3 NADH, 1 FADH2. So 4 dehydrogenase step will be there. So you can see here enzyme name. So there are total 8 steps. You can see the dehydrogenase, isocitrate dehydrogenase. Just remember dehydrogenase. This is dehydrogenase. Then you can see here dehydrogenase. Here also you can see dehydrogenase, and here also you can see dehydrogenase. So how do you memorize the name? So you remember the name in shortcut at least. ICD, KGD, and then SD and MD. You can see. So SD, MD, ICD, KD. If you remember this 4 enzyme name, you already know 4 steps. This step, this step, this step, and this step. 4 steps you already know. You already know that.
And those are the only 4 steps that will likely get on an exam, right?
Yes. These are very, very important steps. And you already know that this is the steps where NADH will be produced in FADH. This is the step where FADH is produced. At all other steps, there will be production of NADH. Because this is dehydrogenase. So once you know the enzyme name, you will know the function also. There will be production of NADH. And in addition to this, there is one more step that you need to memorize. This kinase, can you see? This is a very different type of kinase. This is called thiokinase. This is here, there will be production of ATP in the form of GTP. So you will need to memorize this 4 step and this 1 step. So this 5 step, if you memorize, that is done. So 4 are dehydrogenase and 1 is kinase. In glycolysis, there are 4 kinase plus 1 dehydrogenase. Here opposite, you will see 4 dehydrogenase and 1 kinase. Which kinase? Thiokinase. Okay. So we will talk about a step now. So we can see. So you already know this step. Pyruvate, this all process is happening inside mitochondria. Remember that. Mitochondria, inside cell. Mitochondria. So you can see pyruvate converting into acetyl. You can see acetyl has 2 carbon here. And this is coenzyme is made up of vitamin B5. So acetyl coenzyme, this part 2 carbon will transport to this 4 carbon molecule like this here. So this becomes citrate. Citrate means 3 carboxylic acid. 1, 2, 3. That is why this is also called TCA, tricarboxylic acid cycle, because first step is formation of tricarboxylic citrate, and this is also called citric acid cycle. Citric acid cycle. So this is synthesis of citrate. So the simple name is citrate synthesis. Very easy name is citrate synthesis, the first enzyme, because synthesis of citrate. First step, citrate synthesis. Now, after that, what will happen? There will be aconitase. So what does this aconitase do? This aconitase changes the position of hydroxyl group from 1 to this. So this becomes citrate to isocitrate, similar to citrate.
Sorry, doctor, could you just go over how it went from 2 carbon to 3 carbon?
Which?
How did acetyl-CoA become citrate, like from 2 carbon to 3 carbon? Where did the extra carbon come from?
No, this is not 2 carbon. You can see this is 4 carbon.
Oh, it starts there. Okay.
Okay, 4 carbon. You can see 4 carbon here, and then on this 4 carbon, this 2 carbon adds here. You can see addition of this carbon will be like this here, and then it will be here.
Okay, got it. Thank you.
Got that? So this is what happens. So 2 carbon is added on oxaloacetate. Okay, and this becomes citrate. This is the first step. This isocitrate undergoes decarboxylation reaction, oxidative decarboxylation reaction. But this is not a similar oxidative reaction. This is different type of. You can see removal of carbon dioxide at 3rd step. At 3rd step, removal of carbon dioxide and production of NADH at 3rd step. And 4th step, you will see removal of 2nd carbon dioxide and production of 4. So consecutive production of this NADH and removal of carbon dioxide. So 2 carbon is there, 2 carbon dioxide is removed here. This is done by which enzyme? First is dehydrogenase, again dehydrogenase. Dehydrogenase remove carbon dioxide and NADH. This is also dehydrogenase. isocitrate dehydrogenase, remove carbon dioxide and produce NADH. This is KGD, alpha KGD, alpha ketoglutarate dehydrogenase produce this. And this alpha KGD is, I have already told you, this enzyme is very similar to PDH. This enzyme also have vitamin B1. Mechanism is very similar, B2, B3, lipoic acid, and then B5. So this also have the same thing as PDH, this enzyme. And this is a big enzyme. I will show you the picture, how big it is. If you compare all the enzyme together, if you put all the enzyme together, and if you compare with this enzyme is like almost similar in size when you put all these enzyme together. So this is a very big enzyme, similar to PDH, okay? But isocitrate dehydrogenase is not similar. So here also, PDH does oxidative decarboxylation. You can see oxidative decarboxylation. So understanding of one reaction will make you understand other reaction as well. So this dehydrogenase, this is dehydrogenase. Now, after dehydrogenase, you can see here, this is a alpha ketoglutarate. This is alpha ketoglutarate. There in PDH, pyruvate use, this is a 5 carbon. Now, because of 5 carbon, this is 5 carbon, because of this isocitrate dehydrogenase, carbon dioxide removed, this becomes 4 carbon. Become 4 carbon here. This is 6 carbon. Because of removal of 5 carbon, 4 carbon. It becomes 4 carbon. So 4 carbon is succinyl coenzyme A. This is the step. You can see succinyl synthase or thiokinase, succinyl thiokinase, where you will see production of GTP. And this, after this, succinate is released. And then coenzyme A goes out of the molecule. Here you can see succinate, again removal of 2 hydrogen from this and this, 2 carbon. This result into fumarate, double bond, insertion of double bond here. This is done by succinate dehydrogenase. Now this fumarate with addition of water here, it becomes malate. Again from malate dehydrogenase will remove 1 NAD. If you go through, you will remember easily, because for our dehydrogenase, so you know the function of dehydrogenase?
Yep.
And then one is thiokinase. You know the function of kinase. And this is a very different type of dehydrogenase, where you will see production of
B2.
Yes, B2 is there, and this is production of FADH2. Great. So let's see. You can see enzymes different. Can you see? This is PDH. Can you see? This is alpha ketoglutarate dehydrogenase. So structure of this PDH and alpha, they are very similar. Made up of E1, E2, and E3. E1, E2 at the center, E3. E1 and E3 at the periphery. So this enzyme, if you combine all these enzymes together, even then this alpha ketoglutarate will be bigger than them all combined. So just you don't have to memorize, just I wanted to provide you context so that you will memorize the thing. This alpha ketoglutarate is similar to PDH. Remember that. And the reaction is also same. Where vitamins are same. B1 to B5. Reaction is also oxidative decarboxylation. To oxidative decarboxylation, this oxidative decarboxylation is not equal to PDH. This oxidative decarboxylation is equal to PDH. Okay, remember that. Okay, so I have already told you 4 important enzymes: isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, this succinate dehydrogenase, and malate dehydrogenase. So you can remember ICD, KGD, STMD that way. So decarboxylation happens at these 2 steps. We have already done this, ICD and alpha KGD. And this ATP or GTP, ATP is equivalent to GTP synthesis. It happens at this step. Succinyl coenzyme thiokinase. This actually thiokinase is basically misnomer. You know, thiokinase is misnomer. But for our memory, for our convenience, because kinase is equal to ATP production of consumption. That is why we have used thiokinase. It is also a name, but more appropriate name is synthetase, because this enzyme fall into group of ligase, not transferase. Okay, you can see no phosphorus transfer. There is production of GTP. This is a part of ligase reaction. Can you see? Okay, so you can see irreversible steps are, these 3 steps are reversible. First step, third and fourth step. This 2 dehydrogenase step. This first step, we will talk about this first step at last. So you can see this coenzyme, whenever something, if coenzyme is bound to anything molecule, coenzyme is bound to this coenzyme, this is coenzyme, acetyl coenzyme. Now I'll tell you one thing very important. This bond is very high energy bond. This bond is high energy bond. High energy bond, okay? And that is why this, because when this bond breaks, there will be release of high amount of energy. High energy. That drives reaction, irreversible reaction. That is why this step is irreversible step and rate limiting step. So this is the logic of first step, and then third and fourth. These are rate limiting step. Irreversible step means rate limit. Irreversible step, slow step, and that is why these are rate limiting step. And here regulation happens at these 3 steps. Now, so you can see here. So one very important enzyme. The enzyme name is succinate dehydrogenase. It is nothing but complex 2. This enzyme where FADH2 is produced. This enzyme is nothing but complex 2. Remember that. This is complex 2 of electron transport chain. This is a very important concept. So this is complex 2 of electron transport chain. This is, you can see membrane here. So this is complex 2 of electron transport chain. So important enzymes we have already done. So you can go through this yourself. Alpha ketoglutarate has this. I have already explained this all. And you can see their production of acetyl coenzyme in pyruvate, here succinyl coenzyme. So from pyruvate you can see pyruvate to acetyl coenzyme A, here alpha ketoglutarate to succinyl coenzyme A. Coenzyme A. Here also coenzyme A, production of NADH, release of CO2, here also release of CO2, production of NADH. So both reactions are similar. This is the very important reaction also. So, and now you can see, as in glycolysis, glycolysis is activated by ATP or ADP, low energy, activated by low energy, okay? Inhibited by high energy, because the end product of glycolysis is ATP. So ATP inhibit glycolysis. The same way, the end product of Kreb cycle is ATP, and that is why ATP will inhibit the Kreb cycle. And NADH is also an energy equivalent. So that is why this 2 energy will inhibit. So basically what happens is, this is what is the purpose of Kreb cycle is the production of energy. Purpose of glycolysis is production of energy. So energy inhibits the reaction. This NADH and ATP will inhibit the reaction. Remember, NADH and ATP will inhibit the reaction. All reaction, actually all pathway, NADH, ATP will inhibit glycolysis also. NADH, ATP will inhibit Kreb cycle also. NADH and ATP will inhibit electron transport chain also. Because these are the end product. Product inhibits. Did you get that?
Yep.
Yeah. So product inhibits. Very simple. That is why this ATP and NADH will inhibit, and reactant, actually ADP is basically reactant. If it is a product, it is an indirect reactant. So this reactant will activate the pathway. ADP and AMP are the activator of this glucose oxidation. These are activator of glucose oxidation. When I say glucose oxidation, all three pathway comes into play. I am talking about the cellular respiration of glucose. Glucose oxidation means cellular respiration of glucose. All three pathway: glycolysis, Krebs cycle, and electron transport. Now, so you already know this. You already know.
Sorry, I have a question. Doesn't calcium stimulate alpha glutarate dehydrogenase? Do we need to know that?
So yes, calcium. That is a good question. Calcium directly does not activate this. Okay, calcium indirectly activates this. How? Because you see, calcium activates this. Okay.
Okay,
so see, everything is connected in chain. They are chain. When it activates this, the production of this is high. When this is produced in high quantity, the actually flux of this reactant will be high. This will be high. This will be high. This will be high. And this reactant always, though it activate through feedback or feed forward or not, if reactant is more, this enzyme will catalyze this more. So indirectly, this calcium activate the whole pathway. Get that?
Yes, I thought it stimulated.
No. Directly, it does not directly stimulate.
Okay,
it actually stimulates here. Pyruvate dehydrogenase is a very important, crucial enzyme. It activates this, and because of activation of this calcium by this, see, calcium by this, what happens? This pyruvate level also become down, and you know this actually, if you take the concept of river, as I told you, from glucose to carbon dioxide as river concept. So what will happen? Anything that facilitates the reaction here will actually, any factor that facilitates any rate limiting enzyme will actually
like a motorboat or
facilitate whole pathway. Did you get that?
yeah, it's like a motorboat going down, right? Like, or a yacht, like faster. Yeah.
Yes. So this is how calcium influences the whole actually metabolic process of glucose oxidation in muscle. Okay.